Palynology. Otto Erdmann, a Swedish botanist, pioneered palynology. The term palynology was introduced by Hyde and Williams in 1944 following correspondence with Swedish geologist Ernst Antevs in the pages of Pollen Analysis Circular, one of the first journals devoted to pollen analysis produced by Paul Sears in North America. P.K.K. Nair is considered as the father of Indian palynology. Palynology is the study of plant pollen, spores, and certain microscopic plankton organisms in both living and fossil form. It is associated with plant science as well as with geologic science. Palynology is used in phylogenetic analysis. Palynological features used in plant systems are pollen aperture, pollen shape, pollen wall structure, pollen size, pollen sculpturing, pollen polarity, etc. So, what is a pollen grain? The pollen grains are the male gametophytes of a flowering plant, and it consists of a thick outer covering known as exine and inner thin layer known as intine. This is a cross section of the cell wall of a pollen grain and the spore. The structure of both pteridophyte and bryophyte spores is very much similar to the pollen. The inner layers, the outer layer that is exine, possess sporopollenin. The inner layers are the endospore and exospore. The outer layer, which often peels off, is the perispore. Pollen wall. The exine is composed of sporopollenin, which is a complex and highly resistant biopolymer containing fatty acids, phenyl propanoids, phenolics, and carotenoids. The intine is largely composed of pectin and cellulose. In entomophilous flowers, there will be an oily layer surrounding mature pollen known as pollen kit, composed of lipids and carotenoids. Pollen grains and spores can be preserved for thousands to millions of years and can be used for reconstruction of earlier vegetation and climatic condition due to sporopollenin. This is the pollen wall structure. The outer exine having different layers, the foot and the upper foot layer, the upper columella, then tectum, then outer sculpturing. The diagrammatic representation of pollen grain showing the pollen coat, tectum, bacula, neck sign 1, 2, exintine, endintine, plasma membrane. The exine is further divided into two layers, the outer ectexine, that is sexine and the foot layer, and the inner endexine, which is often well developed in dicots. Exine sculpturing plays an important role in attachment to the pollen insect pollinators. Sculpturing. The sculpturing of exine of pollen grains are important characteristics for identification. The inner structure of exine can be either tectate, or intactate. The sculpturing of pollen grain is ornamentation of the exine surface, which can be ciliate, foveolate, areolate, gemmate, clavate, vericate, baculate, echinate, regulate, striate, or reticulate. These are the different forms of exine surface, and in 3D, three-dimension, they appear like this. The pollen stratification. Then coming to pollen aperture. Pollen aperture is an opening in the pollen grain, colpus or pores. Colpi are regarded as more primitive than pores according to Takhtajan. The number of apertures can be designated by the prefix mono, di, tri, tetra, penta, hexa or poly to the terms colpate or porate. Coming to the next one, that is a pollen unit. It refers to the number of pollen grains united together at the time of release. It may be monad, that is a single pollen unit seen in majority of angiosperms. Example, hibiscus, trionum. Dyad, Ascarina philippinensis. Tetrad, tetrahedral tetrad. Example, Ericaceae. Tetragonal tetrad, example, Fabaceae. Polyad, the example is Parkia speciosa. Then Pollinium, the example for this is members of Apocynaceae and Orchidaceae. Pollen size. The pollen size is measured in terms of both polar diameter and equatorial diameter. It varies tremendously across taxa. Pollen grains are 25 to 50 micrometer, and the shape of the pollen may be spheroidal, oblate, prolate. Pollen polarity. It refers to the position of one or more apertures relative to the spatial reference. Pollen grains from the direction of a pole is known as polar view. Observing from equatorial direction is an equatorial view. The different types are apolar, heteropolar, and isopolar. Pellenogram. A pellenogram is a diagrammatic representation of pollen grain or microspore regarding its symmetry, aperture, shape, size, sporoderm stratification, sex sign, pattern, etc. Thank you.